Good day, everyone. We are Group 10 reporters and we are here to discuss our topic entitled Gene Therapy versus Stem Cell Therapy. I'm the first reporter of our group, John Liberiano, to be followed by Miss Rachel Sabilao and Mariela Grace Pacho. Gene therapy versus stem cell therapy. At the end of this lesson, the learner should be able to
Define what gene is. Differentiate gene therapy from stem cell therapy.
explain the importance of gene therapy in the future,
Demonstrate a process of gene therapy and discuss examples of genetic diseases.
Before we proceed to our discussion, what comes to your mind when you heard the word genes and therapy? As we all know, gene is a basic physical functional unit of heredity. Genes are made up of DNA. Some genes act as instructions to make molecules called proteins. However, many genes do not code for proteins, while therapy, also called treatment, is a process of finding processes for possible cure. So now, what is gene therapy?
Gene therapy is a technique that modifies a person's gene to treat or cure disease. Gene therapies can work by several mechanisms, replacing a disease-causing gene with a healthy copy of the gene.
And what is the stem cell therapy?
Stem cell therapy is a form of regenerative medicine designed to repair damaged cells within the body by reducing inflammation and modulating the immune system. This phenomenon makes a stem cell therapy a viable treatment option for a body.
Understanding gene therapy. The expression of genetic condition is not always as simple as the amount of deoxyribonucleic acid or DNA present or the inheritance patterns of single gene disorder. There are many factors that determine how one's genotype or genetic makeup correlates to much one's phenotype or the physical manifestations. Among these are phenotypes, expressivity, mosaicism, and X inactivation.
Mosaicism occurs when a person has two or more genetically different sets of cells in his or her body. If those abnormal cells begin to outnumber the normal cells, it can lead to disease that can be traced from the cellular level to affected tissue, like skin, the brain, or the other organs. Mosaicism likely occurs on some small, unnoticeable level in most humans. It happens after the sperm fertilizes an egg, forming a zygote, which grows through a process of cells dividing over and over and over. When cells divide and multiply, they make an exact copy of their DNA, and then split the copies between each of the resulting two cells. There is a mistake anywhere in this process. For example, a DNA copy that contains a mistake or DNA that is split unevenly. The resulting cells will be different. In some cases, the abnormal cell may simply die. But if it survives, the result is mosaicism. If this occurs early in development, as many as 50% of a person's cells could be abnormal, which will be a high level of mosaicism. If the mistake happens later, however, a smaller percentage of cells will be abnormal, resulting in a lower level of mosaicism. The impact of mosaicism is typically determined by the severity of the mistake in the DNA, as well as the degree of mosaicism.
Type of disorder caused by mosaicism. Mosaicism can cause many different kinds of disorders, including the following: ichthyosis with confetti. This is a disorder that causes red scale skin all over the body. Klinefelter syndrome. This syndrome can cause low amounts of testosterone. This can lead to problems with sexual development and other issues. Crippel-Trinoni syndrome. This disorder causes a red birthmark called a port wine stain. It also causes excess growth of soft tissues and bones, as well as abnormal veins. Mosaic Down syndrome. Down syndrome, this is a condition causes intellectual disabilities and delays, weak muscles, and flat facial features. It can also cause a heart defect, digestive problems, thyroid problems, and other health issues. Pallister-Killian mosaic syndrome. This is a development disorder that causes weak muscles, intellectual disability, thin hair, patches of abnormal skin color, and other birth defects. Ring chromosomes 14. This condition causes seizures, intellectual disability, and delayed speech and motor development. SOX2 antiphthalmia syndrome. This is a rare disorder that can cause a child to be born without eyes. It can also cause seizures, brain problems, and delayed growth. Triple X syndrome. This condition is caused by an extra X chromosomes in each of women's cells. It can cause learning disabilities, delayed language skills, and problems with motor skills and muscles. Trisomy 18. This condition can cause slow growth in the womb, heart defects, a small head, and other defects. Only a small number of children with this condition live past their first year. Turner syndrome. This is a condition in women that causes short hair, ovarian problems, lack of fertility, and heart defects.
X inactivation. As it turns out, the level of gene activity produced by a single X chromosome is the normal dosage for a human. Men have this dosage because, well, they only have one X chromosome. Women have the same dosage for a different reason. They shut down one of their two X chromosomes in process called X inactivation. In X inactivation, an X chromosome is compacted, or as my intro bio professor liked to say, crumpled up into a ball, to make a small dense structure called a bar body. Most of the genes on the bar body are inactive, meaning that they are not transcribed. Process of X inactivation was discovered by the British geneticist Mary F. Lyon and is sometimes called Lyonization in her honor. Each parent. Which one will she inactivate? X inactivation is a random process that happens separately in individual cells during embryonic development. One cell might shut down the paternal X while its next-door neighbor might shut down the maternal X instead.
All the cells descended from each of these original cells would maintain the same pattern of X inactivation. Example of X inactivation: calico cat. A classic example of X inactivation is seen in cats. If a female cat is heterozygous for black and tan alleles of an coat color gene found on the X, she will inactivate her 2X and thus the 2 alleles of the cat color gene at random in different cells during development. The result of this is a tortoiseshell cat pattern made up of alternating patches of black and tan fur. The black patches come from groups of cells in which the X with the black allele is active, while the tan patches come from the cells in which the X tan allele is active.
Why gene therapy is important. Genes that are flawed and do not work properly can cause disease. Gene therapy is a technique for correcting defective genes responsible for disease development. Researchers may use one of several approaches for correcting faulty genes. A normal gene may be inserted into a non-specific location within the genome to replace a non-functional gene. This approach is most common. An abnormal gene could be swapped for a normal gene through homologous recombination. The abnormal gene can be repaired through selective reverse mutation, which returns the gene to its normal function. The regulation, the degree to which a gene is turned on or off of a particular gene could be altered.
Penetrance is a measure of how often a disease genotype correlates to the disease phenotype. For example, in a completely penetrant disease, every individual who is genetically positive for the disease will have a clinical manifestation of the same disease. In contrast, for disease with incomplete penetrance, some individuals who are genetically positive for the disease may be entirely asymptomatic.
Organisms inherit genetic information in a variety of ways that results in continuity of structure and function between parents and offspring. Suppose you as a child have a genetic disease, one that could be cured by diet or similarly straightforward therapy. Wouldn't the parents want the disease diagnosed? Or take another example. Perhaps two people are getting married. Wouldn't it be beneficial to know what chance their offspring have of inheriting one of several harmful genetic traits? We are now armed with a different advanced procedure like PCR, modern bloating, RFLP mapping, and DNA sequencing.